Okay, welcome to the last lecture of these kind of overview of microbiology lectures in the clinical microbiology course of Path Elective. I'm going to take you through parasitology really quick. This is going to be kind of a broad overview. I'm going to kind of skip a lot of things because we're going to be talking about this a lot later. So the classical definition of a parasite, what is a parasite? They always say that it's an organism that gains benefit from a living host while the host experiences some harm. So this would be sometimes described as a plus minus or a positive negative relationship. In medical microbiology, we're usually talking about a pathogenic eukaryote, a single cell organism, a worm, or a bug. We're not usually talking about a fungus or a bacterium or a virus, even though those usually are parasites. So the different classes of parasites, we have protozoa, helminths, and arthropods. The protozoa can be broken down into amoebozoans, ciliates, flagellates, apicomplexans, and then the helminths can be broken down into nematodes, trematodes, cestodes. The arthropods, you definitely know a lot about these. Lice, body lice, pubic lice, hair lice, or head lice. And we'll talk a little bit more about those as well. There's other arthropod parasites. The protozoans, the first class is the amoebas. So these are actually in the Sarcodina subphylum, and the Sarcodina subphylum, they are the big characteristics they have, that they have a trophozoite, which is a motile reproductive phase, and then they have a cyst, which is an immotile phase, which is enhanced environmental stability. One of the examples of amoebas that we like to use is Entamoeba histolytica. So Entamoeba histolytica can enter the host as the cyst phase and then will transmit through the organism in its trophozoite phase. And so that's where it can get to the brain, it can get to the liver, the lungs, many different places. Another amoebozoan, though, is Naegleria fowleri. So this is one of those brain-eating amoebas that you hear about. This actually has a flagellated phase. So in the environment, it can be flagellated, a trophozoite, or a cyst, but the only infective stage would be the trophozoite. So that trophozoite would be you can think of troph, as like the living form that's getting into the organism, and that's really what's going to spread to the brain because it's going to go up that cribriform plate into the brain, and that's transmitted through kind of aerosolized water. So common parasitic amoebas, we talked about Naegleria fowleri. We also talked about Entamoeba histolytica. There's also Entamoeba dispar and Entamoeba moschkovii. But there's also intestinal amoebas that are symbionts, or at least commensals. So Endolimax nana, Iotamoeba Buchlei, and Blastocystis hominis are actually just the commensals, so they're non-pathogenic. Other tissue-based amoebas that are pretty important are the acanthamoebas and Balamuthia, and so those ones will be in the tissues. I skipped ahead of myself there with Balantidium because that is the next slide. Ciliates, or in the Ciliophora phylum, these are very similar to the amoebas. They'll have a motile stage that's, the reproductive form, and they'll have the cyst phase, which is the more immotile and environmentally stable form. And these are asexually reproducing. The only known ciliate parasite that causes human disease that we know of right now is Balantidium coli. And so the Balantidium coli will have its cysts in water or on food, and that will infect the host, and that will then get into the intestines, and it can have it transform into its trophozoite phase and reproduce. So flagellates are a really important class. So the example we have on the right is Giardia lamblia, Giardia intestinalis, or Giardia duodenalis. So yeah, this is one of the more important intestinal flagellates. So flagellates are broadly broken down into GI/GU flagellates, or gastrointestinal genitourinary flagellates, and then hemoflagellates, or blood/tissue flagellates. So the GI/GU flagellates Very similar to amoebas, they have a trophozoite and a cyst phase. On the right, we're looking at Giardia lamblia, also known as Giardia intestinalis or Giardia duodenalis. They also, so some of these don't have cyst forms though. So cysts are not seen in Trichomonas vaginalis or Dientamoeba fragilis, but that doesn't mean that they're not characterized as a flagellate or a GIGU flagellate. Okay, so the Mastigophora, I forgot to let you know that was the formal phylum name, but the GIGU flagellates on the right, this is an example of the Trichomonas vaginalis, which is a sexually transmitted flagellate, and you can see there's no cyst phase. It doesn't really need it because it doesn't need to be stable in the environment. It goes directly from person to person. So here are some of the examples of the pathogenic GIGU parasites that are flagellates in the Mastigophora phylum. So we have Dientamoeba fragilis, Trichomonas vaginalis, Giardia duodenalis, also known as intestinalis or lamblia. So there's many different names for that. It's not fully worked out yet, that phylum, that genera. So the non-pathogenic examples, we have Trichomonas hominis and Chylomastix mesnii. So those are the examples we have there. The protozoa, the protozoa that are flagellates in the Mastigophora phylum that are in blood/tissue or blood and tissue, these are going to be the hemoflagellates. So hemoflagellates are very different. They'll have a promastigote, an amastigote, an epimastigote, or a tripomastigote. So this it gets a little complicated because different hemoflagellates will have different forms, but hopefully I have a good figure here that I made that hopefully will help you. So I'm going to start in the middle kind of with amastigote. Amastigotes are obligate intracellular cells. So they do not have a flagella, so they're not able to move around, but this is usually where they form a kind of a cyst in tissues. Sometimes they'll call it a tissue cyst. A promastigote will have, a polar flagellum. An epimastigote will have a flagellum that is attached just anterior to its nucleus, and then just posterior to the nucleus would be tripomastigote. So let's look at this kind of broadly. Promastigotes and amastigotes are seen with Leishmania species. Amastigotes, epimastigotes, and tripomastigotes are seen with Trypanosoma cruzi, and then epimastigotes and tripomastigotes are seen with Trypanosoma brucei and other non-cruzi trypanosomes. So here's the figure that I have here. Remember that the sandfly transmits Leishmania, the kissing bug transmits Trypanosoma cruzi, and the tsetse fly transmits Trypanosoma brucei. So let's look at these different forms. Amastigote, aflagellated, T. cruzi, Leishmania. Promastigote, single polar flagella, Leishmania. Not seen in any of the trypanosomas. Okay, so trypanosoma, not going to be promastigote, but that is specifically seen in its vector. The epimastigote is seen in the vector for the trypanosomas. So the epimastigote, you can see it's just here, right next to the nucleus. And it's going to be just, I'd say it's just behind, but it's actually technically anterior to the nucleus. But anyway, so it's just behind the nucleus, anterior technically. And that's Trypanosoma brucei, Trypanosoma cruzi. So that's going to be seen in the insect vector. The tripomastigote is seen with Trypanosoma cruzi and Trypanosoma brucei. So the Trypanosoma cruzi, you can see it's a little bit special because Trypanosoma cruzi can go through the blood. It's transmitted by the vector, but it also can get into tissues and cause some problems. So getting in the tissues can cause something like a cardiomegaly or a megacolon or a megaesophagus. That's where that amastigote comes into play. Here's an example of African trypanosomiasis caused by Trypanosoma brucei. Transmitted by the tsetse fly, gets into the blood. It has the tripomastigote over here, and it has the epimastigote over here. And you can see the epimastigote and the promastigote. And then here is an example of the triatomine bug. And so the triatomine bug is the kissing bug. It transmits Trypanosoma cruzi, gets into the body. This is going to be the trypomastogote. It can form an amastogote in the tissues, go back. It can form another trypomastogote, or it can just be a trypomastogote from previously. Then it can get into the kissing bug again and form an epimastogote. And then the epimastogote can also become a trypomastogote and then get into the humans. So it's a kind of a complicated life cycle. That's something I think you should always look at the CDC website for their life cycles, because they do a really good job. Okay, and then this is the example of the sandfly with leishmania. So you can see the promastogote here, the promastogote, and then the amastogotes here. So the promastogotes in the fly, and then the amastogotes in the human tissues. So apicomplexans, they're complex. They're also some of the most important protozoans and some of the most important pathogens in the world. They have sexual apicomplexans and asexual apicomplexans. So the sexual apicomplexans, they'll have a kind of sporogeny. They'll also have a schizogyny. So the schizogeny is going to be the asexual reproduction, and the sporogeny is going to be the sexual reproduction. Examples of sexual apicomplexans would be Plasmodium, Babesia, Cryptosporidium, Cyclospora, etc., because that's going to keep going on. But the asexual apicomplexans, they'll have a bradyzoite, which is going to be slowly dividing, and they'll be found in tissues, and then a tachyzoite, which will be rapidly dividing and motile. And that example is, of course, Toxoplasma gondii. The tissue cysts for Toxoplasma gondii are most likely going to be made up of the bradyzoites. Okay, apicomplexan cell structure, very different from normal cells. They actually have this odd structure called an apicoplast. That was derived from from like a plant plastid. Doesn't go through any photosynthesis, but it's very similar to a cell structure. So you can actually use that. You can actually use herbicides sometimes to treat these. This is a target for that has been explored as a future a target for therapies. Okay, and then, obviously we have a nucleus, we have these dense granules, and we have these ruptures and the apical complex, which is what it's got its name from. So this is the Morissette lab at UCI that works on the structures of these apicomplexans, and you can see they all, from Plasmodium to Toxoplasma, all have the similar structures. They just have different shapes and forms. Okay, so we're going to talk about helminths very briefly. Helminths are broken up into trematodes, which would be the flukes. So think about like schistosomas. You have liver flukes, lung flukes, intestinal flukes, blood flukes. Then you have cestodes, which would be the tapeworms. Think about fish tapeworm, beef tapeworm, pork tapeworm, dwarf tapeworm, dog tapeworm, etc. And then you have nematodes. The nematodes are going to be the roundworms. So that would be, think about hookworms, threadworms, microfilariae, Loa, lymphatic filariae, and larval migrans. So all those, very important to remember those. So then let's look at the arthropods again very briefly. We have head, body, and crab louse. We have the bed bug, fleas, bot flies, which can cause myiasis, which is when they lay their larvae inside the skin, and then the larvae start to pupate inside the skin and can crawl out. Scabies, which is where the bug kind of burrows into your skin. So these are all kind of examples of arthropods. Different methods of diagnosis. PCR is becoming very popular if you have it available and if you have the time. Microscopic exam of stools, CSF, histology, blood smears. The important thing about microscopic exam of stools, which we're going to talk about later, you definitely have to make sure you look over multiple days because the sensitivity is not that great. Okay, and then immunologic detection. You have ELISAs or enzyme immunoassays. You also have immunohistochemistry, immunofluorescence, and you also have rapid antigen tests. So those are all of those, a very brief overview of parasitology. I hope you guys really like it. Again, I use BioRender for my figures and CDC for a lot of these life cycles. And there's the textbook resource if you need it. Thank you so much.